Good morning, students. So today we will be studying about RDT tools, host cells. Now, host cells are the living system or cells in which recombinant DNA molecule can be propagated or cloned and can also be expressed to produce desired protein. So we can say, as we know, recombinant DNA technology involves isolation of desired gene of interest from one organism and introduction of that foreign DNA into the vector DNA, and thus a recombinant DNA molecule is formed. Now this recombinant DNA molecule is now introduced into the host cell, where that recombinant DNA multiplied and expressed to produce the proteins, right? So the host cells जो है वर्क करते हैं as system for cloning of the foreign DNA as well as expression system. मतलब एक होस्ट सेल के अंदर जो फॉरेन जीन है वो मल्टीप्लाई भी करती है, दैट इज आइडेंटिकल कॉपी बनाती है, इसे हम कहते हैं क्लोनिंग प्रोसेस. और दूसरा जो फॉरेन जीन हमने होस्ट सेल में डाला है वेक्टर के साथ में, वो अपने आप को एक्सप्रेस भी करता है. एक्सप्रेस करने का मीन्स है that the foreign DNA undergoes transcription to produce RNA and then translation to produce proteins. So this means the process of production of protein by the process of transcription and translation from the gene is called as expression of that gene. Now, host cells work as experimental models, means there are number of host cells, prokaryotic as well as eukaryotic cells, which we use for carrying out various experiments, for cloning experiments, for production of various proteins, right? So there are different type of host cells. Prokaryotic cells, mainly bacterial cells. In case of eukaryotes, fungi, animal cells, plant cells, even insect cells, mammalian cells are used as host cells. Now these are the examples of host cells which we are using in genetic engineering. First group is prokaryotic cells. So bacteria is the group, bacterial cells. Or most commonly used bacteria is Escherichia coli, E. coli, and then Bacillus subtilis is also used. Streptomyces species are also used. Then eukaryote, in case of eukaryotes, fungi, animal cells and plants. Among fungi, Saccharomyces cerevisiae, that is the yeast, is most commonly used because it has very simple structure. Then fungi, Aspergillus nidulans is also used. In case of animal cells, we use insect cells, mammalian cells, even oocytes are used for culture. Even the whole small little organisms are also used. In case of plant cell, protoplasts are used either, or complete intact cells are used. Even whole plant is used for genetic engineering, thus to make completely transgenic plant, right? Now, if we take prokaryotic host. So among prokaryotes, E. coli, that is Escherichia coli, this is the first organism which is used in the rDNA technology experiments, because this is very simplest cell. It is a gram-negative bacteria, it is common bacterium of human and animal intestine. So what are the advantages of using E. coli as host cell in rDNA technology? First is E. coli can double in number after every 20 minutes. It has a very short life cycle, quickly multiplies into a large number, right? Thus the bacteria multiply, their plasmid along with the foreign DNA also multiply to produce millions of copies, that is clones of foreign DNA. So if we want to do gene cloning within E. coli, we can get number of copies or clones of foreign DNA within few minutes, and then easy to culture. Very simple nutritional and environmental conditions are required for the growth of E. coli. We can simply easily prepare a broth media or a nutrient agar media and culture plates with E. coli, and within 24 hours we get full plate covered with whole broth filled with that E. coli culture, right? And it has so simple structure, it can be easily manipulated, can be easily rDNA can be easily incorporated into the cell. Then what are the limitations of using E. coli? We cannot use E. coli for every experiment. Why? Because first thing, they produce endotoxins and they are toxic in nature. So they may cause like intestinal infections, they may cause diarrhea. Then they have low exportability of proteins from the cell. So if we are using E. coli for production of any protein, then that protein cannot be easily transported out of the cell. So when अगर हम चाहते हैं that foreign protein का extraction, तो फिर हमें more additional purification steps will be required. Then post translational modification like glycosylation does not take place. तो जो कुछ यूकैरियोटिक प्रोटीन्स होते हैं जिनका प्रोडक्शन अगर हम ई कोलाई में करते हैं तो वो उसके अंदर ई कोलाई में ग्लाइकोसाइलेशन पोस्ट ट्रांसलेशन मॉडिफिकेशंस नहीं होता. दो for some proteins to be completely active and it is very important to have glycosylation. Then proper folding of proteins does not take place. Some of the proteins are like that glycosylation and complete proper folding is protein is required to get the complete active structure of protein. So which cannot take place in E. coli cell. Then one more point, E. coli might degrade recombinant proteins, right? So these are the limitations of E. coli. Although the limitation of degrading of recombinant protein by E. coli can be circumvented, we can use mutant cells of E. coli. हम E. coli के mutant forms use कर सकते हैं which may be defective in production of one or more protease enzyme, so that the recombinant proteins are not degraded. Or we can also use certain mutants E. coli cells जो कि जिनके अंदर over expression of protein folding proteins, that is chaperones का production हम over production करा सकते हैं. But post translational modifications like glycosylation वो E. coli में नहीं हो सकता. So that is why E. coli as a host cells has limited utilization. So कुछ ह्यूमन या मेलियन प्रोटीन्स या फिर हम ये कहेंगे यूकैरियोटिक प्रोटीन्स का प्रोडक्शन हमें यूकैरियोटिक सेल्स में ही होस्ट सेल्स में ही करना पड़ता है. Another prokaryotic cell is organism is Bacillus subtilis. It is rod-shaped non-pathogenic bacteria and therefore it is used as host cell in industry for production of various enzymes, antibiotics, insecticides. इन सब के production के लिए हम use करते हैं Bacillus subtilis को. Then it is used as an alternative to E. coli. Then अब अगर हम बात करें यूकैरियोटिक होस्ट की, so eukaryotic organisms are preferred to produce human proteins because post-translational modifications like glycosylation or phosphorylation, correct disulfide bond formation, proteolytic cleavage, ये सब takes place. All these post-translation modifications takes place in eukaryotic organisms. So they produce stable and biologically active proteins as compared to prokaryotic expression of clone gene. फिर use of eukaryotic host is not associated with generation and interference of toxin. मतलब जो toxins endotoxins जो वो prokaryotic cells बनाते हैं that is not found in case of eukaryotic host. So that is why it is preferred, eukaryotic hosts are preferred for production of proteins and therapeutic agents that are useful for humans and animals. Medicinal value वाले जो proteins हैं थेरेपी में कई यूज आते हैं फॉर ह्यूमंस और एनिमल. इन दैट केस वी जनरली प्रेफर यूकैरियोटिक होस्ट एज कंपेयर टू प्रोकैरियोटिक. देन मोस्ट कॉमनली यूज्ड यूकैरियोटिक होस्ट सेल इज yeast, Saccharomyces cerevisiae. This is most commonly used because it is very simple structure. It is single celled organism which can be easily grown. Its biochemistry, genetics, physiology are quite known. So it is very simple to carry out experiments on that. It has naturally occurring plasmid also. स्ट्रॉन्ग प्रोमोटर्स हैं उस प्लाज्मिड के अंदर, सो दैट अब जो भी जिस भी जीन का हमें एक्सप्रेशन कराना है वी कैन सिंपली पुट इट अंडर दैट control of that promoter in that plasmid and can carry out the expression of that gene. Then it can bring out post-translational modifications in the protein. Then the secreted or recombinant proteins can be easily isolated since very few host proteins are secreted. This means ये सेल के अपने खुद के जो प्रोटीन्स हैं वो बहुत कम बाहर एनवायरनमेंट में, मीन्स जो ब्रॉथ है जिस लिक्विड में या मीडिया में जिसमें हम उसको कल्चर कर रहे हैं उसमें वो सिक्रीट नहीं करता, रादर देन जो रिकॉम्बिनेंट प्रोटीन या फॉरेन प्रोटीन जो उसकी बॉडी में या सेल में जो सिंथेसाइज हो रहे हैं वो सिक्रीट करता है आउटसाइड द सेल. सो इट बिकम्स वेरी इजी टू प्योरीफाई द रिकॉम्बिनेंट प्रोटीन फ्रॉम द होस्ट सेल्फ प्रोटीन्स, क्योंकि रिकॉम्बिनेंट प्रोटीन्स कहाँ विल बी सिक्रीटेड आउटसाइड द सेल. देन यूएस FDA जो है फूड एंड ड्रग एडमिनिस्ट्रेशन जो है उन्होंने एज सेक्रोमाइसिस सेरेविसे को ग्रास ऑर्गेनिज्म डिक्लेयर किया, दैट इज जनरली रिकॉग्नाइज्ड एज सेफ, GRAS, जनरली रिकॉग्नाइज्ड एज सेफ ऑर्गेनिज्म. So it is used in baking and brewing industry from decades. But now biotechnologists are able to use yeast to produce a large number of recombinant proteins like insulin, alpha-1 antitrypsin, hepatitis B virus surface antigen, platelet-derived growth factor, fibroblast growth factor, HIV-1 antigen. So number of these additional value वाले products या diagnostic agents, vaccines जो है we are able to produce inside the yeast cells. But there is again a limitation of yeast cell. A very low or a limited yield is obtained. बहुत कम होता है production. Difficulty in secretion of some proteins and sometimes hyperglycosylation. Like glucose units are attached. That is post-translational modification हो रहा है. But sometimes that is hyper. Excess glycosylation हो जाता है. Then one more, besides Saccharomyces cerevisiae, we are also using yeast, Hansenula polymorpha, for synthesis of alpha and beta globin chains of human hemoglobin. So yeast is the simplest eukaryotic host cell. Then certain fungi like Aspergillus, Trichoderma, Mycelioptera, Thermophila, this is recently being used for gene cloning experiment and have been developed into expression system for screening and production of various industrial enzymes, right? Then mammalian cells. Mammalian cells जो है that are also employed as host cells, mouse cells we are using. Advantage of using mammalian cells यही है कि certain complex proteins which cannot be synthesized by bacteria can be produced in the mammalian cells, like tissue plasminogen activator हुआ. This is mainly because mammalian cells के अंदर complete machinery होती है. Machinery means सारे enzymes and proteins are present, सारे organelles are present, which is not present in prokaryotic cells. So total machinery is there to modify the protein to in its final form. That is, the post-translational modifications can occur in mammalian cells. So the gene manipulation experiment in higher animals and plants are usually carried out to alter the genetic makeup of an organism. मतलब जब हम transgenic plants या transgenic animals बनाना चाहते हैं, we just simply introduce foreign gene into the अगर animal बनाना है then although various techniques है but it can be introduced in the say embryo of the animal and the whole transgenic animal can be produced. Same way plant के अंदर हम even प्रोटोप्लास्ट के अंदर हम इंट्रोड्यूस कर सकते हैं और एनी सेल के अंदर इंट्रोड्यूस कर सकते हैं. We will be studying like agrobacterium tumefaciens जो है TI प्लाज्मिड की मदद से हम ट्रांसजेनिक प्लांट्स बना सकते हैं. So we are using animal cells and even plant cells as host cell in our DNA technology. Right, so I hope आपको ये आज का lecture समझ में आया होगा. Then besides that, also used as a host cell. So we will be discussing this thing in our next lecture. Thank you.